Hi all, Dr. Clark here again, and today for Gen Bio, we're going to talk about Mendel, Gregor Mendel, and his pea experiments, and this is kind of the beginning of genetics. So we're going to talk about what we call Mendelian genetics, and then we're going to progress into what we know now about genetics, which is far more than what Mendel did, but nonetheless, we're going to start with Mendel. So Mendel worked on garden peas, and I'll give you a little bit more of his background in a little bit. But nonetheless, he found out a mathematical method to predict the offspring of two parental crosses. Okay? And that really started the flow of information into figuring out chromosomes and genes and alleles, and eventually the Human Genome Project into creating GMOs and all kinds of things like that. And so it was really his work that kind of paved the way to modern genetics, and he's known as the father of genetics. So with that, let's talk more about Mendel and the garden pea. First of all, we should say that all this research started really in the 1850s, and Mendel did his work in Borneo, okay, which is now I think it was part of the Czech Republic, and now it's Russia. But Mendel was a monk. Okay, an Austrian monk. But at the time, it didn't really matter who you were because no one knew how traits were inherited, how material was passed from parent to offspring. Now, there were guesses. Okay? Charles Darwin had a guess. Remember, this is about the same time period as Darwin. Darwin published his Origin of or Evolution by Means of Natural Selection in 1858. So there was definitely this time period that Mendel and Darwin could have crossed paths, but they didn't. They didn't know each other, at least according to most research. They knew nothing of each other. But Darwin had an explanation for how things were inherited also, which he called genules. And so Darwin in the 1800s thought that, or hypothesized that little teeny pieces, very fine, minute pieces of all your organs and everything that makes you, break off and go down into the gonad region of males and then the gonad region of females, and are passed on. And then the new individual is a combination of those particles. Okay. Now, it was a pretty good idea at the time, but we now know that to be not true. But it was Mendel who came up with how things can be inherited. So heredity is kind of given to Mendel's work with garden peas. So inheriting, or material being passed from parent to offspring, at the time when Mendel was working, he called the features, the heritable features, characteristics. And any difference in the characteristic, he called it a trait. Okay? We now know characteristics to be called genes, and the different types of genes or variations within that gene we call alleles. Okay? But nonetheless, Mendel was definitely on the right path. Okay? And so, again, just like Darwin discovering how species change over time without knowing there was such thing as genetics or DNA or chromosomes or anything like that, Mendel also came up with the way at which we inherit characteristics without knowing there was such thing as DNA or chromosomes or anything like that. And so he did it by mathematics, quantitative science, by counting and measuring. And so we give Mendel the credit for solving the puzzle of heredity. And that's why he's known as the father of genetics. So this is a picture of Gregor Mendel, and like I said, he was an Austrian monk who worked on garden peas, mainly garden peas, and figured out the genetic relationship of garden peas, and mathematically figured out the probability of certain offspring. Now, a couple things about Gregor Mendel that I should put out there. I'm not Mendel's biggest fan, and you'll see why. But his work did pave the way for modern genetics. He gets a lot of credit for doing genetic work, and at the time when he was doing it, it was amazing work. But you'll see there's a lot of changes that we made to Mendel's work, a lot of adaptations, and a lot of people that along the way don't get any credit for modern genetics. But Gregor Mendel made a huge mistake. He made a great discovery, first of all, but then made a huge mistake afterwards. And so his discovery of how traits are inherited was amazing. Okay? He selected the right species. He selected garden peas, which, in my opinion, he got lucky. Okay? He could have chose a lot of other plants like Charles Darwin. Darwin chose roses, which is unlucky, and we'll talk about it later. But Mendel chose peas. And peas have an enormous number of traits that are characterized by single genes. Okay? So he could work out colors of the peas, the pods, the placement of the flower, the shape of the seeds, and all this because they're all controlled by single genes. And he figured this out through manipulations and garden experiments, and figured out mathematically a probability. Now, here's where the blunder comes in, though, at least in the opinion of a scientist like myself. He published his material in the most obscure journals possible, okay? And no one read it. Like, no one read it. It was hidden away in these very obscure journals, and it wasn't read by anyone. Anyone in the field of molecular science, no one read it for about 50 years. So Mendel was publishing in the 1850s, 1860s, and it really wasn't until about 1900 when people figured out that Mendel knew mathematically how to predict offspring. So we went almost 50 years without knowing anything about Mendelian genetics. And then there's a couple grad students that were just digging through old stuff, and they found these journals by Mendel, and sure enough, he'd already figured out what people were still working on for the last 50 years. So that was, in my opinion, that's kind of a blunder. It set genetics back, well, 50 years or so. And we'll talk about another one that occurred more modern time, and not the result of obscure journal, but the result of maybe sexism. But anyways, we'll come to that later. So Mendel, like I said before, he performed his experiments on garden peas, which was a great plant to study. They're super ideal for genetic studies in the sense that, first of all, most of the traits that you can see, so you can distinguish between colors or texture or placement of flowers or height of the plant and these different things. There's a lot of varieties, and most of the material is very suited for genetic work. They're easily grown in a greenhouse. Most people can grow a garden pea very easily from seed to reproductive parts and fairly quickly. Okay? I'm talking maybe a couple months. About 60 days for some varieties, or maybe 3 months for other varieties, maybe 90 days. So all in all, it's an excellent plant. On top of that, this is what makes it a very unique plant. is the flower parts can be manipulated. So inside a flower of a, well, typical flower, you'll have two main parts. You have a female part, which is called the pistil, and so it has ovaries inside in a region, and then a pistil tube with a platform, the stigma. Okay, and pollen lands on that, forms a pollen tube that goes down and fertilizes one of the ova. Okay. Around that pistil is anthers, okay, stamen and anthers are going to be the tops of the stamen, and this is going to be where the pollen, or the male gamete of the plant, which you can just consider sperm, the pollen of the plant is held. Okay. And so a lot of plants have this rejection method where pollen from themselves cannot be placed on the pistil, on the stigma, and have a pollen tube formed. They recognize that as self-pollen, and they can't self-pollinate. But in the case of peas, you can self-pollinate peas, which makes them very unique. So you can cause them to go through asexual reproduction by pollinating themselves, okay, or self-reproduction. The other thing is you can remove the anthers. They're big enough, and you can manipulate things. You can cut all the anthers off. You can pollinate them by just using a paintbrush and moving pollen from this plant to the next one, on and on and on. So they're easily manipulated. And then, like I said, they can self-fertilize, which makes it great for genetic experiments and test cross and things like that. Okay. So Mendel's first experiment was to design what we call true breeding varieties. Okay? So he first had to establish some true breeding varieties. And what a true breeding variety is, every time you self-reproduce that variety, it gives you the exact same character. So whatever character you're interested, from a pea point of view, or in the case of Mendel, from a pea point of view, that was the true breeding variety he had to establish. So if it was like seed color, so maybe the seeds are either green or yellow. And so a true breeding variety would be a plant that produces green seeds constantly or yellow seeds constantly. Okay, and so that's how he had to establish that. So in order to establish that, he had to do crosses and self-fertilization. And so he self-fertilized plants until he ensured that only one type of the trait, or what we call allele, was present. And so it was just constantly the exact same thing over and over and over again. He called those the parental lines, or the P generation. Then he would cross the P generations, the true breeders, together. So if we're talking about purple flowers and white flowers, he would cross those two true breeding varieties to get the alternative traits. He called that the F1 generation, and we still use true breeding P generation, F1 generation in our notation when we talk about these simple crosses. So he'd cross those. That's the F1 generation, the offspring from those parental generation crosses. And then he would self-fertilize the F1 generations to form the F2 generation. And this allowed him to mathematically predict or calculate the probability of producing the varied types of genetic difference in pea plants. So here you can see what I'm talking about. So self-fertilized, he just took pollen from the anther and transferred it to the stigma for this individual. Took pollen from this anther, transferred it to the stigma for that individual. Produced all purple, produced all white. Then he took pollen and he transferred it to this individual, or vice versa. It didn't really matter. Okay? And he gets his F2 or his F1 generation. Okay? Oops, sorry. Then he takes that F1 generation and he crosses it with himself. So again, just pollen from this individual onto the stigma, and then you see the varieties. So this is how he would get the mathematical probability from a cross between what we call now two heterozygote individuals. So what he was observing was, he did this many, many times, and what he observed was that one of the traits from the parental cross to the F1 cross, one of the traits would disappear. You wouldn't see it anymore. So you had two true breeding varieties, you cross them, and the F1 generation you had only one of the characters. And then the F2 generation, that character would reappear. The one that was present in the F1 generation, he called the dominant characteristic. Okay, so now I want you to make sure that you understand. When something is considered dominant, it doesn't mean it's more likely to occur. It doesn't mean that in the population it's more represented. Okay? It just means that when the dominant trait is paired with the recessive trait, the dominant phenotype, okay, which in this case maybe it's flower color, is going to be the one that you see, not the recessive phenotype in these general crosses. The recessive trait is the trait in the F1 generation that's gone and reappears in the F2 generation. So recessive doesn't mean worse, it doesn't mean less common, it doesn't mean anything like that. It just means that in the presence of a dominant trait, the recessive trait will not be expressed. So here is just a table of his experiments. So purple flowers are dominant to white flowers. Okay? And he found the ratio of purple to white to be 3.15 to 1, or almost 3 to 1. Yellow seeds are dominant to green seeds. Now, most of you are like, oh, this is weird, because I only eat green peas. That's the recessive form. See, like I said, it's not a matter of what's common, what's not, okay? But if you take a pollen from a green seed plant and you mix it with pollen from a yellow seed plant, you're going to produce more yellow seeds than you are green seeds. And that's what he did. Again, at a 3 to 1 ratio. Round seeds are dominant to wrinkled seeds. Green pods are dominant to yellow pods. Inflated pods are dominant to constricted pods. Most of us know a constricted pod if you grow. And we also, most of us have white flowered pea plants, green seed pea plants, and constricted pods. Axial flowers, terminal flowers, tall plants, dwarf plants. Okay, so these are the ratios, but look at those ratios. They're almost identical all the way. 2.8, 3.1, 2.9, 2.8, 2.9, 3.0, 3.1. They're almost all 3 to 1 ratios. Mathematically, Mendel was onto something. So what he determined from these simple crosses was that you would come up with a 3 to 1 ratio in the F2 generation if you crossed two true breeding varieties. So the parental line goes to the F1 line, then the F2 line, and it was going to come out as a 3 to 1 ratio. Now later, he discovered that that 3 to 1 ratio is the phenotypic ratio. That's physically what's being presented. But what's really going on is a genotypic ratio of 1 to 2 to 1. So in actuality, okay, what he's seeing is one true breeding dominant plant, two not true breeding dominant plants, and one true breeding recessive plant. Okay, we'll come back to this in just a second. Okay. So here you can see crossing those parental lines, okay, results in an F1 generation where you can determine that purple is dominant. Okay, self-fertilize that purple, or the F1 generation, and you return, now you have a 3-to-1 ratio. But what's going on is you have some, okay, that are 25% that are going to be true breeding dominant, 50% that are going to be not true breeding dominant, and when you cross them, they produce the recessive or the non-dominant variable, and then 25% that are going to be true breeding recessive, always producing the recessive trait. Okay. So from this, Mendel proposed 5 general hypotheses to explain these mathematical results in his plant results, his count. The first hypothesis was that parents do not transmit traits directly to their offspring. So this was in direct regards to, at the time, we had different scientists in the 1800s that were saying that, you know, if you had a certain trait, you could pass that directly to your offspring unchanged. Okay? And those were kind of Lamarckian ideas. Lamarck was a biologist that said that, you know, it's basically inheritance of acquired characteristics could also be possible. So both cases. And later he was, you know, not supported by much evidence at all for really anything. But nonetheless, at the time in the 1800s, he had a lot of people that were following his ideas. Well, Mendel came out with his and suggested, hey, parents do not transmit traits directly to their offspring. And parents transmit things that he called factors, which we now call genes. And so the genes are passed on, but not the actual phenotype, not the actual trait. And we'll see why that's important here in a second. Hypothesis 2 was that each parent contains 2 copies of the factor governing each trait. Now, in this case, that was true for Mendel because he selected what we call a diploid organism, an organism that has 2 copies of every chromosome, 1 copy from the mother line and 1 copy from the dad line or father line. Okay? Now we now know that had he chose something that was not diploid, he would never been able to come with this hypothesis. So you can actually remove the 2 copy piece and you can put, you know, any number there, because we know that there's tetraploid organisms, hexaploid, octoploid, all kinds of varieties, okay? And so that really doesn't change the overall hypothesis, you just got to change these numbers. The numbers don't really work for all organisms, right? And he noted that homozygotes or homologous chromosomes or homozygotes will have the same copy of the genetic material of, in this case, what he called factors, which we now call genes. So if you're a homozygote, you will have the exact same gene on both your chromosomes. Okay? And exact meaning that at least phenotypically it's the same. Okay? So if you're dominant, okay, for both genes, they'll both be dominant. If you're recessive, they'll both be recessive. But if you have one of each type of genes, then you're called a heterozygote or heterozygous. Okay? And that means you might have a dominant from your dad and a recessive from your mother, or vice versa. His third hypothesis is that alternate forms of the factor lead to alternate traits. And what he called traits, we now call alleles. And that's just a alternate form of the gene. So again, like I said, you could be dominant or you can be recessive. Okay? These are alternate forms of the gene. And let's remember, genes are really just genetic code that codes for a certain protein and a certain structure of that protein. Well, you change the genetic code just a little bit, then this structure is going to be a little bit different than this structure. And that's the difference that we get between dominant and recessive. Right? And so you can receive different varieties from each parent, which gives you the genes or the genetics of the individual, which we call genotype. And then we have the expression of those alleles or the genes, which we call phenotype. His fourth hypothesis was that two alleles do not have an effect on each other. So your chromosome number 1, doesn't have an effect on chromosome number 2. We now know that his hypothesis number 4 is incorrect. Okay? And we'll talk about some variations to this. Okay? His hypothesis number 5 was that the presence of an allele does not ensure that the trait will be expressed in individuals that carry it. Okay? So heterozygotes will only show the dominant allele as it's expressed. And we also know that hypothesis number 5 is also not true. So this is just a good piece of evidence that Mendel, when he was coming up with his theory for Mendelian genetics, he had a list of 5 hypotheses, okay, in which was supported by his evidence. But remember, Mendel was only working on one species, the Garden Pea. Okay? And so as other experiments came about, okay, other test subjects came about, we then had to change that Mendelian genetics theory to remove certain hypotheses, like alleles not affecting each other, or the fact that if you're a heterozygote, you're only going to show the dominant form. Okay? Both these are not true, and I'll show you evidence for those both being rejected. But that doesn't change Mendel's theory, except for that we had to remove them and alter his theory by just obtaining 3 hypotheses, and so on. Okay? So with that, I'm going to come back and talk about Mendelian genetics, and we're going to dive into Punnett squares, which some of you are fairly familiar with because you probably had them in high school. And we're going to look at monohybrid or single character cross versus dihybrid, a 2-character cross, and then how there is tri and on and on, quad and penta, and on hybrid cross, where you have multiple characteristics that can be crossed at the same time. Okay? And then from there, we'll look at where Mendelian genetics falls apart, okay, and the players in that role. Okay? So next time, we'll pick up that piece.